Beta-glucuronidase, Wikipedia audio. Beta-glucuronidases are members of the glycosidase family of enzymes that catalyze breakdown of complex carbohydrates. Human beta-glucuronidase is a type of glucuronidase that catalyzes hydrolysis of beta-d-glucuronic acid residues from the non-reducing end of mucopolysaccharides such as heparan sulfate. Human beta-glucuronidase is located in the lysosome. In the gut, brush border beta-glucuronidase converts conjugated bilirubin to the unconjugated form for reabsorption. Beta-glucuronidase is also present in breast milk, which contributes to neonatal jaundice. The protein is encoded by the GUSB gene. Human beta-glucuronidase is synthesized as an 80-keta monomer before proteolysis removes 18 amino acids from the C-terminal end to form a 78-keta monomer. Beta-glucuronidase exists as a 332-keta homotetramer. Beta-glucuronidase contains several notable structural formations, including a type of beta barrel known as a jelly roll barrel and a Tim barrel. Human beta-glucuronidase is homologous to the Escherichia coli enzyme beta-galactosidase. This homologous relationship, along with the knowledge that glycosidases often perform hydrolysis catalyzed by two acidic residues, enabled the development of a mechanistic hypothesis. This hypothesis proposes that the two glutamic acid residues GLU540 and GLU451 are the nucleophilic and acidic residues, respectively, and that the tyrosine residue TYR504 is also involved in catalysis. In support of this hypothesis, experimental mutations in any of these three residues result in large decreases of enzymatic activity. Increased activity of an E451A mutant enzyme after addition of azide is consistent with GLU451 as the acid/base residue. Using analysis of labeled beta-glucuronidase peptides after hydrolysis of a substrate that enters a very stable intermediate stage, researchers have determined that GLU540 is the nucleophilic residue. Structure. Though the particular type of nucleophilic substitution employed by beta-glucuronidase is unclear, evidence for the mechanisms of their homologs in the glycosidase family suggests that these reactions are qualitatively SN2 reactions. The reactions proceed through a transition state with oxocarbenium ion characteristics. Initially, these mechanisms, because of this oxocarbenium characteristic of the transition state, were suggested to be SN1 reactions proceeding through a discrete oxocarbenium ion intermediate. However, more recent evidence suggests that these oxocarbenium ion states have lifetimes of 10 femtoseconds, 0.1 nanoseconds. These lifetimes are too short to assign to a reaction intermediate. From this evidence, it appears that these reactions, while having an SN1 appearance due to the oxocarbenium ion characteristics of their transition states, must be qualitatively SN2 reactions. The specific activity of TYR504 in the catalytic mechanism is unclear. Through comparison to the structural data of the homologous enzyme xylanase, it has been suggested that TYR504 of beta-glucuronidase might stabilize the leaving nucleophile or modulate its activity. Molecular graphics images were produced using the UCSF Chimera package from the Resource for Biocomputing, Visualization, and Informatics at the University of California, San Francisco. In addition to these residues, a conserved asparagine residue has been suggested to stabilize the substrate through the action of a hydrogen bond at the 2-hydroxyl group of the sugar substrate. Deficiencies in beta-glucuronidase result in the non-recessive inherited metabolic disease known as SLY syndrome or mucopolysaccharidosis 7. A deficiency in this enzyme results in the buildup of non-hydrolyzed mucopolysaccharides in the patient. This disease can be extremely debilitating for the patient or can result in hydrops fetalis prior to birth. In addition, mental retardation, short stature, coarse facial features, spinal abnormalities, and enlargement of liver and spleen are observed in surviving patients. This disease has been modeled in a strain of mice as well as a family of dogs. More recently, researchers have discovered a feline family that exhibits deficiencies in beta-glucuronidase activity. The source of this reduction of activity has been identified as an E351K mutation. GLU351 is conserved in mammalian species, which suggests an important function for this residue. Examination of the human X-ray crystal structure suggests that this residue, which is buried deep within the TIM-barrel domain, may be important for stabilization of the tertiary structure of the enzyme. In the crystal structure, it appears that ARG216, a member of the jelly roll domain of the protein, forms a salt bridge with GLU352, therefore, GLU352 is likely involved in stabilizing the interaction between two different three-dimensional domains of the enzyme. In molecular biology, beta-glucuronidase is used as a reporter gene to monitor gene expression in mammalian and plant cells. Monitoring beta-glucuronidase activity through the use of a GUS assay allows determination of the spatial and temporal expression of the gene in question. Mechanism of catalysis. SLY syndrome. Molecular applications, use as a reporter gene.